Okay, thank you, and thanks, Ted. Some really interesting work on predator-prey dynamics that I think will really sort of shape how we think about cod recovery in Maine. I'm going to go on to probably the most low-tech talk for the session. We're going to hear about some high-tech solutions from the next speakers. But I'm going to talk to you about what we can learn just by taking pictures from fish. So that might include just taking pictures from a simple iPhone or any kind of point and shoot. The answer is quite surprising, what we can find out. So when we think about modern fishery science, we often think about these complicated methods and these expensive equipment. And I've been accused of using a lot of this in the past. But I'm not going to talk about any of this stuff, this lab stuff, and this field stuff today. I'm just going to be talking about cameras. And high-tech is great for sure, especially for focused questions or very well-funded studies. But sometimes low-tech is desirable, particularly for exploratory work and for putting data collection in the hands of many. So this can include citizen science, where you can just get lots and lots of data from lots and lots of people, and taking pictures is one way to do that. Another example, in the case of water quality monitoring, this is perhaps, there hasn't been more powerful low-tech solution than a simple dinner plate. So some of you might be familiar with the Secchi disc, which was invented about 150 years ago. And with this particular device, which is just a black and white device that you lower in the water column, you can measure water clarity, and many studies correlating that to water quality have been conducted throughout the world in both oceans and lakes. For fish work, we can do something that requires even less time and effort, and that is taking pictures. So again, with something as simple as an iPhone or a point and shoot, we can collect lots and lots of pictures of fish. With pictures, we can assess body shape, which can say a lot about a fish's lifestyle. For example, whether it's more sedentary or migratory, and also where it's from. So in other words, we can look into population structure. So how does this work? So genetics and lifestyle can lead to very subtle differences in the relative proportions of various measurements in the fish. So in this example, we have a cod, and we have those little white dots are called landmarks. And from those landmarks, we can construct what's called a box truss design, and then take all of those measurements and use it. And this is where it decidedly gets less simple. It's more complicated in the lab. So I won't dwell too much on the statistics of this, just to say that we employ multivariate statistics to test for differences in body shape among groups, or we can actually use this analysis to suggest groups. So even if we don't have any a priori idea of what groupings should be, the statistics can often guide us in this direction. I should just reiterate that a lot of these differences I'm going to talk about are not really revealing to the naked eye. They're very subtle and can usually only be picked up by this type of analysis. So what have we learned using this approach? So I'm going to give a couple examples, or 3 examples, in the Gulf of Maine. The first is red cod. Some of you who fish cod might know that they come in different colors. There's the usual sort of drab gray-green color, but sometimes we see very red colored cod. And so we've done a lot of analysis to look at how lifestyle differs between red cod and normal cod. We've also looked at how closed areas may select for resident cod, and some of this actually draws on our body shape analysis. And also looking at population structure. This is a big question for cod in the Gulf of Maine, throughout the Northeast, and also related to recovery. We need to know, or at least we need to, I think, act more on what we know about population structure. So in the first example, we have red cod, and here's just a picture of what I was talking about. This coloration could be vastly different. These are 2 cod caught on the same line, so there were 3 hooks. And we brought up those 2 cod on the same line at Cash's Ledge. So we know that there's lots of red cod coexisting at Cash's Ledge with these normal cod. And so the question is, are these color differences just fleeting, related to diet, or is color indicative of an entirely different lifestyle for cod? So we conducted that body shape analysis. We took all those measurements, which was 18 different length measurements, and then we sort of boiled it down into the statistical analysis. And what this graph shows, and I won't get into it too detailed, but what it shows is that the red cod there on the left have different body shapes than the normal cod over there on the right. And we can actually, sorry, before I get into that, there's actually a sort of a functional difference to this difference in body shape. If you look on the left, those points actually correspond with less streamlined bodies, so they have shorter heads and deeper bodies. And those points on the right correspond with more streamlined bodies, and so those are longer heads and shallower bodies. So if you think about migratory capacity, you'd think of a fish that has to swim long distance ought to be more streamlined. It would tend to have a more shorter head and a shallower body than a fish that would be sticking around, might have a deeper body and sort of more compressed this way. And so that's what actually plays out right here, and that's what's driving these differences in shape. When we then go to see how well we're doing in terms of statistical analysis, we can actually reclassify the fish back into their respective groups, that is red cod and normal cod, 85% of the time. So even if we didn't know anything about color at the outset, if we just did the body shape analysis, 85% of the time we would be able to say that one fish is red and one fish is not. And so that's actually a pretty high reclassification success rate. So what we found then is that red coloration goes with a more sedentary life style, and just basically what this means is that red cod are not built for long distance swimming like other cod might be. And acoustic work at Cash's Ledge has actually confirmed this residency hypothesis. So we've put acoustic tags on these fish at Cash's Ledge. We've set up an acoustic array to watch their movements, and those red cod really stick around a lot more than the normal cod that we've also tagged. Okay, the next example is looking at closed area effects on cod life history. So in 2007 and 2008, we sampled inside and outside of these four major closed areas in Gulf of Maine and Georges Bank. And we found that growth, feeding, and age was enhanced inside closed areas, and probably the most significant result was that we found a consistent effect on age. So there was older cod inside of closed areas compared to outside. Each of the paired closed area pairs here. And we found that as a whole, there were 8 times more old cod, that is older than 5 years old, inside closed areas than outside closed areas. So that was a major finding for us. And it's something that doesn't get considered a lot when we talk about the value of closed areas. This idea of demographics, this idea of maintaining old fish in the population, and closed areas seem to be doing that. But aside from that, we were also interested in body shape, and that is, do closed areas select for more stay-at-home cod? So we just thought that this might be something that would play out just because you're creating these large areas, and cod do show this variation in behavior that we know of, and so it would stand to reason then that we might actually be selecting for those cod that are staying at home, staying inside these closed areas. So we conducted the analysis, and so again, we boiled down all of those body shape measurements into, in this case, one variable, which is the variable along the y-axis there, that's mean score, DF1. And then we looked at all of the 4 different closed areas, and then the dark bars are inside and the white bars are outside. And you can see it's paired design. And in every case, the body shape was more streamlined outside of closed areas than inside of closed areas. So what this suggests then is sort of what we hypothesized, is that the closed areas tend to harbor these fish that have these body shapes that are consistent with not swimming as much, not migrating as much as the fish that you find outside of closed areas. So that was interesting. And so this sort of suggests then that cod inside closed areas have body shapes less adapted for swimming. So this is just one of those, another, sort of not fisheries-induced selection, which we hear a lot about in terms of age of maturity, size of maturity, but this is a fishing management-induced selection on cod life histories. So closed areas may be selecting for these more resident cod, but I think what this also suggests is that cod are taking advantage of closed areas to stay put. So this is not something, body shape is not something you change overnight, it's something that you grow into, and the fish that we're finding in there, they've been there for long enough to change their body shape, and that suggests also that they're there, they're resident, they're using the areas, and we could say that closed areas may be having an effect that way. Okay, the final example, I should say number 3, is population structure. So actually this is a summary of the the slide that Ed just showed with all those release and recapture locations for the tagging study headed by Shelley Talek a number of years ago. And so what she and others have concluded is that well, we have our Gulf of Maine stock and our Georges Bank stock, and if we live in a perfect world, those should line up with population structure. But in reality, what we find is that there's a fair bit of movement between particularly the western Gulf of Maine and western Georges Bank. There also appears to be sort of a distinct group out of eastern Georges, which appears to interact with the Bay of Fundy. And genetics work has also supported this dichotomy. So we did the same type of thing. We boiled down all of those body shape measurements, and we came up with 3 groupings. Now there's a lot of overlap here, and you would expect this because you look at the overlap that's suggested by tagging as well. But in any case, we see some of these groups pulling out in different directions. And what was neat about this is that if you look to the right side there, that big circle, it incorporates Jeffrey's Ledge, so that's western Gulf of Maine, and closed area 1, western Georges, so they're identical. But where the differences occur are eastern Georges, what we expect from tagging, as well as Cash's Ledge, which is central Gulf of Maine, which doesn't show up in the tagging. And so that suggests a third group, this central Gulf of Maine group. I'll just jump through quickly. So this is how it would line up spatially. We have those ellipses up there. We have our western component, which straddles the stock boundary. We have our eastern component of Georges. And then we have this third group, the central Gulf of Maine. And more photos will hopefully sort of address the question whether those mid-coast and eastern Maine fish are part of that central Gulf of Maine group as well. Just to expand a little bit on that, in the Gulf of Maine, if we look at one of these body shape variables, again, boiled down from all of those measurements into one variable, you can actually see a break in that variable around 69.6 degrees latitude, which corresponds basically to Platt's Bank. So it runs right down the middle of Platt's Bank. So on the west side of Platt's Bank, you get one body shape. On the east side, you get another body shape. And what's interesting about this is that it corresponds very well with Ted Ames' previous analysis suggesting a break between the western Gulf of Maine subpopulation and the mid-coast Maine subpopulation. So there's some interesting agreement there. So just to wrap that up, body shape can provide powerful insights into the secret lives of fish. Body shape provides an easy test of an hypothesis, so example, resident versus migrant, or population structure. That can be followed up with other studies. So it's not to say that body shape is sort of the answer in and of itself. Usually it's kind of point you in a direction, and then you can follow up with other studies like tagging or genetics. And I think the real power in body shape analysis is that the data is really easy to collect. We can get tons and tons of pictures and we can just pour through them. So thank you.